which is on genomes. All right, well, we've been talking a lot about genes, both genes from the Mendelian standpoint and the structure of genes like introns and exons and how they're grouped into these operons in bacteria and things like that. But we haven't really used this word genome yet. Genome means all the genes that you find inside an organism, all the genes that it has. But it also includes all the DNA that they have that's not genes. It essentially means all the DNA, whether it's genes or not genes. Here you see some cells there, which are, each cell has its nucleus stained. So all the genetic material in there, all the DNA, is the genome. Here's a close-up of some human chromosomes. And of course, each chromosome is a big piece of double-stranded DNA with hundreds or thousands of genes on it. Now, you may wonder what I mean by this, by other DNA. What the heck does that mean? Well, if you look at the DNA along any given chromosome, segments of that chromosome's DNA are going to be genes, meaning that they encode proteins. But there are some other segments of the chromosome which is still G's and A's and T's and C's, but it does not encode any proteins. And you might wonder, well, why do we have DNA on our chromosomes if it's not protein encoding? Well, hold that thought. We'll discuss what this non-protein encoding DNA is all about a little bit later on in this genomes lecture. But anyway, all the DNA, whether it's encoding proteins or not, is considered part of the genome. And each cell has the entire genome. Each and every cell in your body, you have trillions of cells, and each one has all your chromosomes and therefore all of your genes, which for human beings, remember, is around 25,000 genes. And for different organisms, their genomes might have more or fewer genes than we human beings do. So if each cell has all of your 25,000 genes, it's kind of an interesting fact that not each cell expresses all 25,000 genes. Some genes are not expressed. They're silent within certain cells, but those same genes might be expressed in different cells in your body. And that ability of a cell to regulate its gene expression, meaning to express certain genes but not express other genes in its genome, is called differential gene expression. And that's what a lot of today's lecture is going to be about. Before we get into how cells can turn on certain genes and turn off other genes within their genome, I want to start by introducing why they would want to turn some genes on and turn other genes off. Well, one of the major reasons is something called cell-specific gene expression. There are many different cell types within your body, and for certain genes, it's appropriate that those genes get expressed in certain cell types, but not in other cell types. Just to give an example here, there are liver cells, there are 3 types of blood cells. There's red blood cells, white blood cells, and a type called platelets. There's neurons, there's muscle cells, and there's actually at least 3 subcategories of muscle cells. There's a type of cell called epithelial cell, and those come in different subvarieties. I remember reading somewhere that there are around 200 different cell types in our bodies, and certain genes they all need to express, like the genes for breaking down glucose for energy. Yeah, every cell is going to need that, but other genes are expressed only in certain cell types. Here's a picture of a woman showing some of the different tissues and cell types in her body. Just to give an example, here in her brain you find neurons, nervous tissue, and one of the things that neurons can do is generate electrical signals. That's how neurons send messages to each other. And so the genes for that would be appropriate for neurons to express, but not other types of cells in your body. These are a picture of the cells in her heart, her cardiac muscle cells. Cardiac muscle cells have some genes that allow them to contract rhythmically to pump the blood. But it wouldn't make sense for your fat cells to be going, blah, 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 blah. That'd be kind of weird looking. Wouldn't do any good. It'd be a waste of energy. So that would be an example of cell-specific expression just in heart cells. This is supposed to be your stomach, and stomach cells express some genes that allow them to make acids and digestive enzymes. But only stomach cells should express those. Imagine if your fingers were making acids. Every time you tried to shake hands with somebody, they'd go, "Blah!" and they would burn up and things like that. No fun. And there are liver-specific genes and leg muscle-specific genes. Every cell has its own particular genes that it's supposed to be expressing. And you don't have to know this exact number, but somebody estimated that every cell is only expressing about 20% of its 25,000 genes, which, what does that come out at? Around 5,000. Only about 5,000 of the 25,000 genes are being expressed in any particular cell. But which 20% varies from one cell type to another is the point here. And they don't know exactly how cells differentiate as we develop. Because if you think about it, we all started off as one single little cell, a zygote. And then that divided, and then that divided, and we became little embryos. And so at first, all the cells look the same. But pretty early on in embryonic development, they start differentiating, meaning they start deciding what type of cell they're going to be. So some of them start to become muscle cells, and some become nervous cells, and some become liver cells very early on. And they don't know how that happens, but it definitely happens very on in early embryonic development. Which leads me to yet another example of differential gene expression. Some genes are only expressed, are developmentally regulated, meaning they should only be expressed at certain times in an organism's lifetime. And a good example of that is the hemoglobin gene, which I guess we were just talking about in terms of sickle cell anemia. Hemoglobin is this protein that's found inside red blood cells. It has a very important role of carrying oxygen. And even a fetus, before it's born, has circulating blood with red blood cells in it because its tissues need oxygen just like our tissues need oxygen. But kind of interestingly, the gene it uses to make its hemoglobin is different from the hemoglobin gene that we people who've been born use. In other words, it's expressing a different type of hemoglobin gene than we do. Matter of fact, the one that embryos use is even a different one than the one that fetuses use. Embryos are earlier offspring stage, and then the embryo eventually grows into a fetus. So there's actually several different hemoglobin genes that are differentially expressed as the embryo develops to a fetus, to a newborn. And you may wonder, well, why is that necessary? Well, there's an interesting physiological reason behind it. The embryo and the fetus don't load up their hemoglobin with oxygen from the air the way we do. They get their oxygen from the mother's blood. And so they need hemoglobin with a stronger affinity for oxygen because their hemoglobin has to take oxygen away from the hemoglobin, from the oxygen in the mother's blood. So you need a stronger attraction to oxygen than regular hemoglobin that you find in adults. Fascinating. And lastly, there's another aspect of differential gene expression is environmentally regulated genes. Some genes might not normally be expressed, but when certain molecules or certain changes in the organism's environment come along, then it might be appropriate to express those genes. Maybe a good example of that is you think of some animals change seasonally. The color of their feathers or fur might change from one season to the next, or maybe they have different behaviors during a mating season and some other seasons. There are genes for those that change due to environmental clues, like if the days are getting longer or shorter, if it's winter, if it's summer, that sort of thing. And in certain cases, there are certain molecules in the environment that might trigger certain gene expression. A good example of that is some of the genes in your stomach lining that express certain digestive enzymes. So your stomach makes digestive enzymes and acids, but it wouldn't be efficient if you were constantly making those digestive enzymes and acids even when you didn't have any food in your stomach. And so only when you put those food molecules in your stomach, only when you eat, do the genes of your digestive system cells start expressing the genes for digestive enzymes. And kind of interestingly, they are very specialized. If you eat a carbohydrate-rich meal, like the corn here, then carbohydrate digesting enzymes get more expressed. And if you eat a fat-rich meal, then fat digesting enzymes get more expressed. Those genes get more expressed. And if you eat a protein-rich meal, then protein-specific digesting enzyme type genes get expressed. So, go ahead.
How is it when a baby is born, when they're inside the womb, they're breathing, well, their lungs aren't breathing oxygen, they're filled with fluid, right?
Yeah.
And then when they're born, the fluid is evacuated from their lungs and they're able to breathe. So when they're genes, is there something that's going on that's like instant when they're born?
Oh, how do they switch from one hemoglobin to another?
Yeah.
I guess I don't exactly know how it works. I suspect as the fetus gets closer to being born, the genes for adult hemoglobin start to be expressed more and more, and the genes for fetal hemoglobin start to be expressed less and less. But it could be even after the baby's a newborn, maybe there's still some fetal hemoglobin around for a while. It's a good question. I don't exactly know. But eventually it does all switch over to the adult hemoglobin form. Alrighty. Anyhow, so the big picture here was that each of your cells has all 25,000 genes, but for different cell types and different developmental stages of your life and different environmental clues, some of those genes will be turned on and some of those genes will be turned off. And that's the nature of differential gene expression. So that's the why. Here's the how of differential gene expression. It's almost always done at the transcriptional initiation level. And what I mean by that is, if you remember this diagram, there are many steps in gene expression. You start off, it's the transcription step, making a pre-mRNA from the gene, and then there's the RNA processing step where the introns are removed, and you add the 5' cap and the poly A tail, and then you have the mature mRNA, then there's the exiting the nucleus step, and then there's the binding to the ribosome step, and then there's the ribosome assembling the protein step, which is called the translation step. Each of those steps could potentially be an area where the cell could control how much of the protein is made, the gene expression. But the major regulation is there at the transcriptional level. If the cell wants to express the gene, it makes the transcript. If it doesn't want to express the gene, it doesn't make the transcript. And it sort of makes intuitive sense. If you're going to regulate gene expression, it would seem kind of wasteful to regulate it here at the translation step. I mean, why go through the hassle of making the mRNA and editing it and sending it out into the cytoplasm and binding the ribosome to it to only all of a sudden at that step not make the protein? Why not just nip it in the bud and not express it there? So let's focus for a moment on the initiation of transcription step. I showed you this before. The blue lines are supposed to be the double-stranded DNA on a chromosome, and there's the first codon of the gene, the start codon, and somewhere down here would be the stop codon. Upstream of the first codon, there's this region called the promoter, where there are certain sequences, promoter sequences like a ta-ta box and a cat box and other promoter sequences. Remember, those promoter sequences are necessary to get the RNA polymerase II to attach onto the template strand down there. Matter of fact, let's kind of zoom in on the promoter there. Here I've got a ta-ta box in the promoter, and somewhere in here there also might be a cat box and other promoter sequences, but I'll just use this to represent the promoter. All right, so when a cell wants to initiate transcription of a gene, to begin to express the gene, at that spot on the chromosome, the two DNA strands move a little bit apart from each other to allow the RNA polymerase to come inside. Here's the RNA polymerase. And I'm not showing it in this cartoon, but there are actually enzymes that are designed to temporarily move the two strands apart to begin the transcription step. Now, remember something interesting about RNA polymerase II. by itself, it's not able to attach to the template strand to make the mRNA. It has no binding site for any sequences there in the promoter. Other proteins need to attach it to the template strand, and those other proteins are called transcription factors. These transcription factors do have binding sites for the TATA box and the CAT box and the other promoter sequences. So the transcription factors come in first. There they are, and bind to the promoter sequences. And then once the transcription factors are on, then they can reach out and grab the RNA polymerase II enzyme and attach it to the template strand. Quick. And then once the RNA polymerase II has been attached to the template strand, then it can run along it like a train on a railroad, making the messenger RNA, which is always complementary to the template strand. Okay, that I think you know so far. That's we've covered that already. But what I didn't mention so far is this. What they found is that the promoter sequences, like the TATA box and the CAT box, and the transcription factors by themselves